Good morning. First of all, let me thank you, Global Student Network, for inviting me and all of you for your participation today. My name is Małgorzata. I work at Poznań University of Medical Sciences. As you can see on the screen, our today's topic is Prion nature of alpha-synuclein, challenges or opportunities. Parkinson disease and related research on diagnostic and therapeutic solutions are in the center of my research work. So let's start with the presentation. According to WHO, the global Parkinson disease mortality rate, the direct cause of death is usually pneumonia, will be dramatically grow until 2016, since available therapeutics only treat the symptoms, and there is also a lack of effective diagnostic method. And diagnosis now of Parkinson's disease is mainly made based on clinical symptoms. Thus, the discovery of early diagnostics and disease-modifying therapeutic now are highly desirable. What is Parkinson's disease? Parkinson's disease, as you probably know, is the most common neurodegenerative diseases after Alzheimer's disease, and clinically characterized by motor dysfunction such as bradykinesia, rigidity, tremor at rest, and postural instability. Patients also represent non-motor manifestations such as olfactory impairment, pain, anatomic dysfunction, impairment sleep, fatigue, and behavior changes. Specifically, olfactory impairment and constipation are very important symptoms, manifestation, which precede the onset of motor symptoms by years. The pathological hallmark in the Parkinson's disease patients in the brain is the presence of Lewy bodies, intraneuronal inclusions, mainly composed mainly of misfolded and aggregated alpha-synuclein. Interestingly, the analysis of samples collected even 20 years before the onset of Parkinson's disease revealed the presence of alpha-synuclein inclusions without almost the whole gastrointestinal tract. The inclusions were detected in submalingural gland, esophagus, stomach, colon, appendix, and many other places. The inclusions were localized in the nerves of entering nervous system. Recently, the controversial a few years ago hypothesis of the prion-like mechanism of alpha-synuclein spreading now seems to have gaining significance as more and more evidence appear. 2 years ago, we searched the PubMed database using keywords like gut-brain axis in Parkinson's diseases and prion-like and Parkinson's diseases, and we noticed the massive, as you can see, the massive increase in the number of publications in these topics, where we gathered and discussed critically in the published review. Now the number is, as you can see, even 10 times higher. The theory of prion-like spreading of alpha-synuclein in the central nervous system was triggered by post-mortem studies in the brains of Parkinson's disease patients who were subjected transplantation with human embryonic midbrain cells. And in the brains of these patients, even 12 or even 22 years after the neurosurgery, Lewy bodies, as you can see, Lewy bodies were present again in the transplanted cells, suggesting the infectious nature of originally present Lewy bodies. In this moment, I should refer to Stanley Prisoner Prion theory, which were formulated many years ago. It was 1982. And what the theory states, according to this theory, infectious protein called, according to the author of this theory, SCARP or transmissible forms or just prion protein can self-proliferate despite the lack of nucleic acid. As you can see on the diagram, native protein, which is presented here as the black circles, as a result of direct contact with prion, its prion forms, presented here as a blue square or a red triangle, takes the relevant, after the direct contact, takes the relevant prion form. Yeah. Regarding Parkinson's disease, alpha-synuclein with beta sheets structure tends to aggregate while the alpha helical structure is a native form. The prion-like spreading mechanism implies that the transmission of the infectious proteins between cells, and it takes in few stages. First is, of course, generation. The generation regarding sporadic form of Parkinson's disease is triggered by external agent, I mean chemical or biological agent, which triggers first event of misfolding protein. In the second stage, the prion protein is released and then translocated between cells, uptaken by the second cells, and within which replication takes place in the mechanism which I showed you in the previous slide. And the process can be only stopped when the prion form of protein is degraded, for example, during the autophagy, which is the subject of different therapeutic approaches which are currently developed, tested. Alpha-synuclein is, as I told you, alpha-synuclein native form is soluble. However, it can adopt beta sheet structure, which have the prion property, which goes that it aggregate into oligomers and then fibrils, and finally into Lewy bodies, which are the cause of cellular regarding Parkinson, there are mainly dopaminergic neurons. As we review, the first event of alpha-synuclein misfolding takes place in the gut. There are in the review which I refer now, we gather a lot of evidence coming from in vitro, in vivo, and even some clinical studies supported the theory. Specifically, the misfolding event takes place in the nerves of entering nervous system or peripheral autonomic nervous system through direct interaction with some environment agent or pathogen. After ingestion or indirectly by dysbiosis. Then the pathology invades the central nervous system in prion-like manner via the vagus nerve. Parallelly, the pathological conditions, I mean the dysbiosis, pathological conditions in the in gut microbiota initiate impairment of gastrointestinal mucosal barrier. Sorry. I'm sorry. So increase the intestinal permeability that cause that bacterial metabolites, some pro-inflammatory cytokines can diffuse to the systemic circulation, triggering inflammatory response, which finally can result in neuroinflammations. As you can see, gut dysbiosis was shown, demonstrated that correlate with increase of putative pro-inflammatory bacteria, increased intestinal permeability, α-synuclein immunoreactivity, and decreased pool of short-chain fatty acids. Small intestinal bacterial overgrowth was usually correlate with increase of motor complication as measured by unified rating, Parkinson disease rating system. Dysbiosis, in this moment I would like to stress the dysbiosis, which is, as I mentioned, very critical condition which can contribute, which probably contribute to the first event of α-synuclein misfolding. And studies revealed that Parkinson disease patients, what is this dysbiosis in Parkinson disease patients? It is related to the decreased in Prevotolace and Lactospiraceae species, as compared, you can see, as compared to the control individuals, and decrease of Lactobacilliaceae and Streptococcaceae, which even in the control, in health subject individuals are very are expressed in very low. I mean that the pool of this bacteria is very low. So the decrease in Parkinson disease patients could be very serious in this case. As I mentioned previously, the α-synuclein pathology invades the brain. That is supported also by large epidemiological studies showing that truncular vagotomy, this is a surgical procedure removing division of the main trunk of the vagus nerve to treat, it was used many years ago to treat chronic duodenal or gastric ulcers, is associated, what is important, is associated with a 40 and 50% decreased risk of Parkinson disease as compared to general population. So all this evidence support the presence of the gut microbiota axis, which is an integrative system of bidirectional communication between the brain and the gut intestinal tract, consisting of central nervous system, enteric nervous system, as well as endocrine, metabolic, mainly short chain fatty acids, which can directly diffuse to the brain, and immune signaling pathways. As the good microbiota or other dysbiosis can contribute to neurodegeneration in Parkinson's disease, strategies based on the modulation of the gut microbiota, including fecal microbiota transplantation, as you can see on the clinical trials group, are already developed and the studies are registered. Another approach is neuroprotection through metabolites generated by intestinal microbiota, such as urolithin A, which is generated from allergic tannins present in the, for example, pomegranate, but also in walnut, wines, and many kinds of berries. The microbiota transformation cause the transformation of allergic tannins into urolithins, especially urolithin A, which was subject of our previous studies. What is important can be distributed to the brain. And we also presented neuroprotective effect of pomegranate juice against Parkinson's disease in a rat model, which we refer to the presence of the urolithin A in the brain. Targeting α-synuclein is also supported by encouraging result of a clinical trial on antibody prasinezumab, which was designed to block cell-to-cell, you can see, which can block cell-to-cell transmission of the aggregated α-synuclein. Now it is in phase 2B PADOVA, that prasinezumab actually is clinically developed in phase 2B. In the context of the targeting spreading of α-synuclein, nanomaterials, especially graphene materials, seems to be very promising approach. Simulation studies showed, as you can see on the screen, that α-synuclein amyloid is affected by different graphene monolayers, as you can compare to the aggregation without monolayers. So it seems that the graphene monolayers vary, especially this one, affect the aggregation of α-synuclein. Kim et al. Demonstrated that incubation of prefibral alpha-synuclein fibrils and synthetic fibrils with graphene quantum dots decrease the number of their fragments beginning from day 3, you can see here, and alpha-synuclein as compared to the alpha-synuclein without graphene quantum dots, which were no longer detectable at 7th day. A recent study demonstrates also that selenium-containing surface induce also destructurization of amyloid samples of alpha-synuclein. Taking into account the direct activity of graphene nanomaterials to direct affect protein aggregation, as I show you, especially α-synuclein aggregation. Dual functionality of graphene nanoparticles are also considered by combining its direct activity with the large surface and the possibility of conjugating the graphene nanomaterials, which made these nanomaterials suitable nanoscaffold for holding very valuable treatment in Parkinson's disease, such as different small molecule drugs, but also small interfering DNA, RNA genes, antibodies, or neurotrophic factors. The improving self-proliferation, differentiation, inference properties of graphene can be also combined with delivery function, for example, in gene therapy or dopamine replacement therapy. Regarding diagnostic, the self-propagation of the pathological form of α-synuclein is already developed to use in novel methods for detection. It's very low concentration based on amplification methods such as protein misfolding cycle amplification, combined with real-time quaking induced conversion. Assay for the early detection of Parkinson's disease in cerebrospinal fluid, which were described last year by was described last year, and seems that the methods is very promising to detect very early Parkinson's disease at very early stage, where in using other methods the low concentration of aggregated α-synuclein in clinical specimen is normally not detectable. SYN1 test is the first commercially available test to accurately detect and visualize, as you can see, abnormal α-synuclein in cutaneous nerve fibers collected from skin biopsy. So this is the first diagnostic approach which can be managed to detect the disease in a non-invasive way. So what conclusion can we draw from this? First of all, the prion nature of α-synuclein, and when we consider its beginning of the disease outside the central nervous system, provide the opportunity of early diagnosis outside the central nervous system, which is more convenient and treat and diagnosis and treatment of synucleinopathy. The second conclusion is as I show you that dysbiosis is one of the suggested pathomechanisms of Parkinson's disease. Fecal microbiota transplantation or neuroprotection could be also developed, which can treat or protect against Parkinson's disease in very early stage. And other therapeutic options such as immunotherapy and bifunctional therapeutics, which targets α-synuclein spreading within cells, are also already developed. So thank you very much for attending.